This recording is going to cover the transport of oxygen and carbon dioxide in the blood. Now, the carbon dioxide and oxygen are gases that have limited solubility, and which could be a problem because our peripheral tissues need a lot more oxygen and they generate more carbon dioxide that can actually just be physically dissolved in your plasma. So the problem is going to be solved by the red blood cell. So the red blood cell will allow us to remove more of the gases from the plasma and bind to them or somehow alter them into something like, when we talk about carbon dioxide, say bicarbonate. And when they do this, by removing them from solution, the gases will all be able to continually diffuse, and we never reach equilibrium. Now, the reactions are reversible. So like if the red blood cell is picking up oxygen in the lungs, at the level of the tissue, it's going to release that oxygen. Carbon dioxide is the same way. At the level of the lungs, we're actually releasing carbon dioxide, but the tissues, we're going to pick it up. So what is always going to determine what direction these go would be the concentration differences between the two. Now, what I do want to mention is the ways in which the gases are transported. And for oxygen, 97% of it is bound to hemoglobin. So there's your role of that red blood cell. Only 3% of it is actually physically dissolved in plasma. But what is dissolved in plasma are the ones that when we ever express a partial pressure of a gas. So if they say PaO2, they're referring to the partial pressure of oxygen in the arterial blood, and that's what's physically dissolved. Now, once the oxygen binds to hemoglobin, it no longer exerts a partial pressure. Now, carbon dioxide actually is transported three ways. 70% of it will be actually transported as bicarbonate. The red blood cell be instrumental in converting carbon dioxide to bicarbonate. 23% of carbon dioxide is transported as carbaminohemoglobin, in which the carbon dioxide is bound to an amino group in the hemoglobin. And then that leaves us 7% that's dissolved in the plasma. So these two here can show you the role of the red blood cell in transport of carbon dioxide. What we're going to do first is we're going to look at the transport of oxygen. Now, the red blood cell contains hemoglobin, and the hemoglobin molecule has it's what we call a respiratory pigment that increases our oxygen carrying capacity about 65 to 70 times, which is tremendous. So we can transport a lot more oxygen because of hemoglobin. The structure of hemoglobin is that we have 4 protein chains called globin chains, and in the center of them we have these heme rings, and each of the heme rings have iron, and it's oxygen that's actually bound to the iron in the heme ring. The globin chains, the protein chains, will be important in one aspect, that it kind of keeps the iron from being exposed to an aqueous environment, which could affect the oxidation state of iron. And another aspect of having hemoglobin the way it is, you have these 4 subunits, is we're going to see how hemoglobin, because of the structure that it has, it can attain different shapes, which can affect its affinity for oxygen, which will be important in transport. So I'm going to show you this curve that's called the oxygen hemoglobin saturation curve. It shows us the relationship between the partial pressure of oxygen here and how saturated hemoglobin is. Now, hemoglobin, if I come back here, can bind up to 4 oxygen molecules. If all 4 are bound, it's 100% saturated. If there's only 3, it's 75% saturated, okay? So if we come over here and you look at this curve, what you'll notice is as the oxygen levels increase, the partial pressure of oxygen levels increase, hemoglobin saturation increases. But it's kind of got a weird shape. It has a little sigmoidal S-shaped curve to it. What it kind of tells you is if you look down here where the slope is fairly steep, is that if I change the partial pressure of oxygen just a little bit, you'll see there's a huge change in how much that we can saturate hemoglobin. It so happens because of the shape of the hemoglobin molecule, its sequence, which determines its shape, is if one oxygen binds, there's a change in shape, a change in conformation that makes it even easier for another oxygen molecule to bind. And when that second one binds, then it makes it even easier for the third one. And so that's kind of an important aspect of hemoglobin, is that it's easy to add oxygen to it. But if you look at the other aspect, is you'll notice that if, we'll go up here first, is at the levels of oxygen you have up in the alveoli, hemoglobin is almost 100% saturated. It's not quite 100%. We never really reach 100% completely because we have some variants of hemoglobin out there, so it's never like completely 100%, but it's pretty close. But what you'll notice is the partial pressure is dropping quite a bit, but look at the saturation didn't change that much. So say, look at it's 60 millimeters of mercury, it's still 90% saturated. That's actually kind of important in that if you think about it, is we can have near normal oxygen transport despite a decrease in oxygen content of the alveolar air. So if you're at high altitudes, the partial pressure of oxygen alveolar air can be significantly less, get down close to 60. You still have a really good transport of oxygen as hemoglobin, and that's where you want most of the oxygen we transport is bound to hemoglobin. That's very important. Or also you think about if you have a condition that significantly reduced your ventilation, which can affect your oxygen levels in the alveoli. If we didn't have hemoglobin around that could still be pretty well saturated even at those levels, that you'd be fatal. So that's kind of good. But then you notice you look down here, you'll see it start to drop. So we can look at this way, it's a sharp curve. At the levels of oxygen that we'd have, say, of the tissues under normal conditions, resting conditions, it's about 40 millimeters of mercury. Hemoglobin is still about 75% saturated. That's pretty good. But that was enough under certain conditions. I just need to release one oxygen per hemoglobin molecule. It's still pretty well saturated. But notice if the levels drop lower, steep drop in that slope, that when the partial pressure starts to get too low, hemoglobin is going to unload its oxygen because that's its purpose. It needs to transport the oxygen and let it go when it's needed. So it's going to release that oxygen. It's going to be, prove it's a very good oxygen reserve. But what we'll also notice is because of the hemoglobin molecule being the way it is, or I should say in having those globin chains and having 4 subunits to it, we can have things that can affect its affinity for oxygen, that such as hydrogen ions and carbon dioxide and some other things that can affect its transport, or I should say more of its delivery of oxygen. So if you look here, it's showing you 2 different things, the effective temperature and the effective pH. Now normal pH, about 7.4, okay? So partial pressure of oxygen level of the tissues is about 40. Here is hemoglobin saturation, okay? Right here. But notice what happens if the pH drops to 7.2. We're becoming more acidic. At the same partial pressure of oxygen, what happened to the saturation? Hemoglobin saturation decreased, which means it delivered more oxygen. It released more oxygen in the tissues. If you see the pH increases, the saturation greater doesn't release as much. So this is the way I want you to remember it is this one, because this will make more sense, is if the pH drops, becomes more acidic, hemoglobin's going to release more oxygen to the tissues. And the way you're going to remember it is think exercise. Exercise is the greatest thing to use as examples. When you're exercising, you may be producing more lactic acid. You want to be delivering more oxygen to tissues because you want to work aerobically. It's telling you I need some more oxygen to make more ATP. So hemoglobin is going to release that oxygen. Now at the level of the lungs, we're going to show you there's going to be a reaction that takes place at the level of lungs is the pH actually will increase because we're going to be getting rid of those hydrogen ions, and I'm going to have a higher affinity for oxygen. That's what you want at the lungs. You want to be able to pick up the oxygen. You'll also notice the effects of temperature. As temperature increases, so here's about normal. If temperature increases, what happens is at the same level of partial pressure of oxygen, look at the saturation. Same as you see before, as temperature increases, hemoglobin saturation decreases, which means you've released more oxygen. So I want you to think about what the effects of exercise. Working muscles are going to be generating more heat. You want to release more oxygen. When you get to the lungs, it's not going to be as hot. We're going to, this case, take in oxygen. You're going to be getting rid of carbon dioxide. Now we're going to look at some couple other things, but before we do that, I do want to mention the effect of carbon dioxide levels and pH, again, on oxygen transport. Is here, well, actually this is kind of showing a similar thing with what you'll notice is a drop in pH, an increase in temperature, increase in CO2, and we'll talk about this one in a moment, an increase in something called 2,3-BPG. These all cause that hemoglobin saturation curve to shift to the right. So what it's telling you, for the same partial pressure of oxygen, you're going to have less saturation. So if you shift the curve to the right, that means the hemoglobin affinity for oxygen is decreased. You're going to deliver more oxygen. This is what we want. Say during exercise, you're going to have an increase in temperature, you're going to have a decrease in pH, and you're going to be producing a lot more carbon dioxide. Now the carbon dioxide needs to be transported by the red blood cell. So thing is, carbon dioxide binds to the red blood cell, and that causes a conformation change. Now it doesn't bind to the same place as oxygen does, but it binds within the globin chain, and when it binds to it, it causes a conformation change in it, changing the affinity of that heme ring for oxygen, allowing us to release it. Now, if you look here, there's a thing called the Bohr effect. The Bohr effect is a very important concept in regards to transport of oxygen. What it tells you is the relationship between carbon dioxide levels and hydrogen ions on oxygen transport. Okay, so it's the effects of carbon dioxide and hydrogen ions on oxygen transport. If you're at the level of the tissues, okay, in the tissues where you're working, they're producing carbon dioxide. They may be producing more hydrogen ions. Because of that increased CO2 and hydrogen ions, its effects on the hemoglobin molecule, that curve gets shifted to the right, our oxygen affinity decreases. So we're releasing more. That's what we want. We need to deliver oxygen more to the tissues. Now, at the level of the lungs, that curve shifts back to the left, because at the lungs, what's going to happen is carbon dioxide is going to be exhaled, and what we're going to show you briefly is hydrogen ions are now going to be out of the question. It's going to be, we'll show you the equation again, is the hydrogen ion levels are going to decrease, the CO2 levels will decrease, so the curve gets shifted back to the left. That allows us to have increased oxygen binding at the lungs. And just think about that's logical. When you're working out, you want to get oxygen in, and the hemoglobin better pick it up at the level of lungs, but when you get to the tissue, you want to release it. So hemoglobin is going to, its job is to transport that oxygen, not to hold on to it for dear life. And when it needs to, it'll let it go. Actually, let's go back to this 2,3-BPG, and then I'll look at the fetal hemoglobin. There's a thing called 2,3-bisphosphoglycerate. Some physiologists like to call it 2,3-diphosphoglycerate. Both of them, bi means two. Is this in glycolysis And the intermediate in the glycolytic pathway is 1,3-BPG. So it just tells you where the phosphate's located. One's located in carbon 1, one's located in carbon 3. Well, you notice this is very similar, except we have moved a phosphate to a 2 position. So this is just a little side reaction that can take place in the red blood cell. And 2,3-BPG, what it does is it shifts the curve to the right. It binds to hemoglobin, again, within the globin chain. It binds to it, causes the affinity of hemoglobin for oxygen to decrease. And so what's so significant about this? Well, the 2,3-BPG levels can increase in response to T3, it can response to growth hormone, testosterone, epinephrine. Those are some of the things that it can respond to. If your blood pH gets a little bit higher, 2,3-BPG levels go up. So what that's going to do is going to improve oxygen delivery to tissues. One way you can think about it is like, say, growth hormone, testosterone. Think like for athletes that that improves their be able to work better because they have improved oxygen delivery to tissues. Now, one thing to keep in mind is 2,3-BPG levels do decline as a red blood cell ages. It does. And actually, I don't know if they still do it, but in the past they used to do this, is blood banks will actually measure the levels of the 2,3-BPG in the samples to determine whether or not to throw out the blood. Because we want to be careful of it, because if it's the red blood cells are too old, they're not going to be able to transport oxygen as much. So they want to make sure that if they're going to do a blood transfusion, because when you bank your blood, it's not good forever. Because as they age, they're not going to be as effective. Now, 2,3-BPG is somehow involved in the differences between fetal and adult hemoglobin. So fetal hemoglobin is different. They do not have the same structure as adult hemoglobin. Is the fetal hemoglobin, though, if you look here, so we're just going to say at partial pressure 40 millimeters of mercury, adult hemoglobin is about 75% saturated. Same partial pressure, fetal hemoglobin is much more saturated. So what's so important about that? Now, it's structurally different than the maternal hemoglobin, and the reason why we have it is it has a higher affinity for oxygen. So it's going to ensure that that baby is going to get oxygen from its mother, because the mother always going to get first dibs on the oxygen. And when that oxygen's going into through the placenta, is that no matter what the mother's doing, she's exercising, it's going to make sure they can grab onto that oxygen from the mom. Now, after you're born, the fetal hemoglobin levels decline and you'll see more adult hemoglobin. Some people, there's a certain level of fetal hemoglobin that persists, but it's structurally different. And then also, its affinity, its ability to bind to 2,3-BPG is different, which ultimately leads to that increased oxygen affinity. Fetal hemoglobin binds to 2,3-BPG less, doesn't have quite the ability to bind to it as much. It comes down to certain amino acid changes. So with a decreased ability to bind to 2,3-BPG, it ultimately has a higher affinity for oxygen. Okay, so that's an adaptation that's needed during fetal development. Now, another thing that could affect your oxygen transport is carbon monoxide. Carbon monoxide results from incomplete combustion of fossil fuels. So if fireplaces, gas engines, those type of things, anything run by gas, that can result in carbon monoxide. Now, carbon monoxide binds to oxygen, or not oxygen, it binds to hemoglobin, the same spot as oxygen, and it binds to that heme ring with a much stronger affinity. So it's 250 times stronger than oxygen, and you have to have really high concentrations of oxygen to kind of get that out of the way. And so what happens is with carbon monoxide is your oxygen carrying capacity of your blood will be severely diminished. And so you're gonna suffer the consequences of low oxygen in your blood. So hypoxia is going to result from it. The weird thing is, though, if you observe a patient with carbon monoxide poisoning, they're not going to look blue. They're going to look pink, because if you do pulse oximetry on the patient, is the oxygen levels dissolved in plasma are fine, but what's the problem is their total oxygen content, their blood is affected because the oxygen bound to hemoglobin is what's significantly affected. So generally the treatment for carbon monoxide poisoning is to give the patient very high partial pressure of oxygen, supplement with some carbon dioxide, because carbon dioxide will help to stimulate them to breathe. Now the last thing we want to talk about is the transport of carbon dioxide in the blood. Now carbon dioxide is transported in 3 ways. So you got dissolved, 7%, 23% is carried through the bloodstream bound to hemoglobin, is carbaminohemoglobin, and then we have bicarbonate, okay? So what happens in the tissues is carbon dioxide is produced by your cellular respiration. It will go into the red blood cell, combine with water to form carbonic acid. You've seen this reaction before. And then it dissociates under the influence of carbon anhydrase to release hydrogen ions and bicarbonate. Now, in the lungs, we're going to see this reaction is going to be reversed, but we're going to not look at that at this moment. What you have to notice is the hydrogen ions are going to be, actually let's talk about hydrogen ions yet, is the bicarbonate will diffuse out of the red blood cell and be transported, physically dissolved in the plasma. But because we're losing a negative charged substance, what we'll see is a chloride will come in exchange for it. They call that chloride shift. That's the term they refer to it as, this chloride shift. The reason for that is to maintain membrane potential, because you've got the negative charges. So this helps to maintain ionic balance between the red blood cells and plasma. But we'll see this reaction will be reversible. Now the hydrogen ions need to be buffered. They combine with hemoglobin. Hemoglobin is a pretty good buffer. They'll bind with it. But that hydrogen ion combining with the hemoglobin will also influence oxygen transport. So we had mentioned that earlier. So it's going to allow us to release more oxygen to the tissues. Now the carbon dioxide that's bound to hemoglobin is carbaminohemoglobin. It's just physically attached to the hemoglobin. And then we do have some of it that's dissolved in the plasma. Now when we get to the lungs, you're going to see everything kind of reverse. So in the lungs, you're going to have oxygen binding to the hemoglobin. And what that does is going to cause the hydrogen ions to be displaced from the hemoglobin. It's going to be released, and it's going to combine with bicarbonate, okay? So it combines with the bicarbonate. Now, bicarbonate goes in, and now we're going to see chloride going back out. So we're just going to keep that negative charge, everything good there. So the bicarb combines with hydrogen ions. You form your carbonic acid. Then that dissociates into carbon dioxide and water. So this reaction is freely reversible. It all comes down to at what point do you have more of something. So at the level of the lungs, we're going to go kind of this way. At the level of the tissues, we go in the opposite direction. And what we have is carbon dioxide is going to be then released from the red blood cell, diffuse in the plasma. It will now become dissolved. And as long as the partial pressure of CO2 is greater here than here, CO2 is going to go into the alveolus. Now you also exhale some water. That's all you do. Just breathe on your hand. Your breath is moist is because water is also released. Now the oxygen binding to hemoglobin also causes that carbon dioxide that was bound to the amino groups in hemoglobin, and that will be released, get into the plasma, and then again that will go across the respiratory membrane into the alveolus, and then again you got your dissolved. So as long as the partial pressure of carbon dioxide is greater here than here, it will travel into the alveolus. So carbon dioxide, you see, is actually transported 3 different ways. Okay, but that bicarbonate though is actually kind of important. Is we can actually use some of that bicarbonate to act as a buffer system. So when we talk about acid-base buffers, we'll talk about bicarbonate again there, and the role of the lungs in acid-base balance. Now that's the remaining part of gas transport. The webinar that follows this would be the regulation of respiration.